Hi, I'm Jake. I'm a fourth-year PhD student at Penn, and I'm going to be presenting my poster about exploring the functional consequences of cancer driver mutations. So generally, the prediction problem that we're interested in, the idea is that we're going to be predicting binary labels that are either the presence or absence of a mutation in a given gene, typically a cancer driver. And we're going to use omics data from TCGA to do this. So this could be something like RNA-seq, this could be DNA methylation data or something like that. So typically it's going to be sort of a samples by genes or features matrix. And so why do we want to do that? So the idea is that we can use this to learn about the information content of a set of genes. So if lots of genes are sort of mutations in lots of genes are well predictable from this data, then we know that that set of genes has a lot of sort of cancer-related information content. Or for a particular omics data set. So if we can use gene expression to predict mutations in known cancer drivers or DNA methylation, then we know that that omics type contains a lot of information about sort of function in cancer. So we can use this as sort of a benchmark in that way. So in general, our experimental design is we're going to take 85 cancer genes from this Vogelstein et al. Paper. So a lot of them are known to be sort of pan-cancer driver genes that are frequently mutated. We're going to do 2 replicates of 4-fold cross-validation that's stratified by cancer type. So the train and test sets have the same proportions of each cancer type in TCGA. And that gives us a distribution of 8 total performance measurements for each omics type or each gene. And the main metric that we look at here is area under the precision recall curve. The idea being that a lot of mutations are sort of imbalanced. They don't occur that often. So using this metric works well with imbalanced labels. And we're interested in comparing against a permuted baseline. So when we permute the labels, that sort of gives us a baseline, a lower baseline that we can compare against, and also between different omics types. So say if you compare DNA methylation with gene expression, which of those has the most information content for a particular gene? So this plot shows sort of aggregate performance over all the 85 genes, comparing gene expression to the 2 methylation array types in TCGA, so 27K array and 450K array. And this sort of shows that on aggregate, gene expression provides maybe slightly better performance for predicting sort of general cancer gene mutations than the methylation data sets, but not that much better, right? And also the methylation data sets provide approximately the same amount of information. And so what this is actually plotting is the mean performance difference sort of for each gene versus the baseline. And the idea there is to correct for label imbalance. So if you have lots of a given label, then the sort of baseline predictor will do well. So it corrects for that. So now what if we add sort of all the data types in TCGA? So we can add RPPA, which is a proteomics data type, microRNA, and somatic mutational signatures to sort of see how much relevant information is in those data sets. And we see that expression and methylation generally tends to significantly outperform all of those data types. So the expression and methylation data sets tend to sort of contain the most relevant functional information. So that was an interesting result. And another thing we wanted to do is we wanted to say sort of, rather than just looking at each data set or each omics type sort of in isolation, what if you combine multiple omics data types? So we took the expression and methylation data since it had the most relevant information. And we took either pairwise combinations of those 2 data sets or all 3 data sets and sort of squashed them together into one simple multiomics model. The idea being to see if we could get better performance that way. And if you look at these distributions, these are 6 sort of well-known driver genes. You can see that for a few of them, so EGFR and KRAS, for instance, you see slightly better performance with the multiomics models. But in none of these cases are the pairwise differences actually significant for a paired sample rank-based test. So this was sort of surprising to us. We thought we would see quite considerably better performance when we sort of squish omics types together, but we don't really see that, surprisingly. So just to sort of summarize the takeaways here, we saw in general that gene expression and DNA methylation across a bunch of different cancer genes tend to sort of contain the most functional information about the consequences of mutations in cancer. And surprisingly, expression and methylation encode a lot of the same information. So if you replace expression with methylation, it doesn't tend to be a huge detriment in terms of performance. So that could be an interesting result for the design of certain studies. We also looked at these multiomics models, and we saw that in general, there wasn't a huge discernible performance improvement over over a model just using single omics data types. Again, the method that we used here was pretty simple. I think there's a lot of room for improvement in terms of more complex multiomics methods. But we saw that for a very simple model, we didn't see much of a performance improvement. So that was a bit surprising. So here are links to our paper and code. Here's my contact information here, and I'd love to talk more offline about this work. Thanks.